So, welcome you all again. Today, in last lecture, we have started about the protoplast culture. So, protoplast, it is nothing but the plant cell without cell wall. So, how this cell that will be, or how the protoplast will prepare that have, or the technique uh, that will be used for the isolations of the protoplast we have discussed in last lecture. So there are two methods from that the protoplast of the plant tissue that will be separated. So first method is the mechanical process and second one is the enzymatic process. So in mechanical process, with the help of some scalpels or under the microscope, the cell wall of the plant cells that will be removed out, and with the help of hand we can easily remove this cell wall of the plant cell in the mechanical process. Uh, this in mechanical process, the plant cells, first of all, the leaf, select the leaf and then peel off the leaves. Then this part or the peels that will be placed into the hypertonic solution. Hypertonic solution, that's why because this hypertonic solution, they will be helping the plant cells for the plasmolysis. So in plasmolysis, the plasmolysis, that part, there is the shrinkage or contractions of the protoplast. So due to the contraction and shrinkages of this protoplast, the cells will be shrinked out, and we can easily remove out the cell wall, that is outer layer of this cells. So in mechanical process, there some merits or demerits are there. So in merits, it is quick method and less expensive. There is no any need of the special solutions or any special instrument. In laboratory, we can easily do this mechanical process. But there have also some demerits because only skillful person they can do this. The new person they are without practicing it is not possible. Sometimes the cell wall that will be breaking out, or it this mechanical method that will be affected to the cells. And also there is also one demerit, that is these cells or protoplast we cannot be stored. If we remove this, then we can immediately they can be used. Otherwise they will be dried out. They are not good in conditions. Then the second method, which is enzymatic process of isolation of protoplast from the plant tissues. So in this method, by using the enzymes, we can remove the cell wall of the plant cell. So in the enzymatic process, there are again two methods: single enzymatic method and double enzymatic method. Single, that means in that method only single enzyme is used for removing this plant cell wall. In double, there is the two different enzymes they will be used for removing the cell wall. So in first, the single enzymatic method, first of all, select the leaf, wash that leaf, and also the wash this leaf with the sterile distilled water. Then again, this leaf will be sterilized with the help of with the help of 10% ethanol for 1 minute, or 20% potassium chromate for the 10 minutes. These two enzymes that will be used for the sterilizations of the leaf. Then after the sterilization, then remove or peel off the epidermis of the leaf. The peel of that leaf that will be removed. Then cut this mesophyll tissue into the fine segment. Then जो अंदर के जो cells है that will be cut out in different segments Then they will be carry out the plasmolysis. Plasmolysis, that means plasmolysis of mesophiles, that means the shrinkage of that cells will be takes place, and the shrinkage will be started when we kept these tissues on the hypertonic solution. So after this wash of the plasmolyzed mesophil tissues with sterile distilled water, and these enzymatic incubations of the plasmolyte tissue is carried out for 24 hours at 25 degrees centigrade. So these mesophyll tissues that will be kept in enzymatic solution up to the 24 hours at the temperature will be 25 degrees centigrade, and this process that will be repeated out 2 to 3 times. Then the enzyme mixture that will be used in this process, that is macroenzyme, cellulase enzyme, or mannitols and s orbital, and their pH is in between the 5 to 4 will be used. So here the mixtures of the uh, enzymes like cellulase, mannitose, and S orbital. The three enzymes mixture, they have been used in this process, and the enzyme having the pH in between the 5 to 4 range. Then these solutions, or they carry out the filtration so as to the separate lip segment debris and protoplasm containing the cellular mixture, they will be separate out. Again, this filtrate or centrifuge this filtrate and to get the cellulate of protoplast. So for only getting the protoplast, again these mixture that will be centrifuged. This process that will be repeated out, which enzymatic incubation to get maximum number of protoplast. जो भी plants के अंदर जो भी protoplast cells है, वो सब प्रोटोप्लास्ट सेल सेपरेट होने के लिए वो प्रोसेस हम रिपीट करेंगे. Then this separated or isolated protoplast that will be stored out in zero point 0.4 molar sucrose solution at 0 degree centigrade. So we can store this protoplast in 0 degree centigrade into the sucrose solution. Sucrose solution in having the zero point 0.4 molar concentration. So this is the single enzymatic method. In that, we have to use the mixtures of the three enzymes, cellulase, then mannitol, and S orbital. So, by using this mixture, we can easily remove out the all materials which are present in the cell. Only we can clearly separate out the protoplast, and this protoplast we can store it in the sucrose solution at 0 degree centigrade. And at the use of these protoplast, we can just remove it from uh, these temperature or in sucrose solution, and they can use directly for practical purposes. So this is the first or single enzymatic method. Then second one is the double enzymatic process. So in double enzymatic process, there are in process you have to see in the flow diagram, there is the enzyme 1 and enzyme second with the different pH grades are used for the separating the protoplast. So in the double enzymatic process, same as the initial process that will be same, that is select the leaf, then these leaf will be sterilized in distilled water, then some chemicals. After sterilization, the peeling off of the leaf will be takes place, then mesophyll tissues that will be cut off in small segment. Then these lip segment, they will be plasmolyzed, means the shrinkages or the contractions of the cell wall or protoplast will be takes place. So we can easily remove out these cell wall. Then this material or the protoplast that will be washing off. So we can easily separate out the protoplast. Then this protoplast again they will be treated with the enzyme 1. Enzyme in enzyme first there is a mixture of the microenzymes uh, and kept this material for incubation for 24 hours or more than. So here in enzyme first the mannitol with pH range 5.8 will be used. Then again the second enzyme that is mixtures of the cellulase and mannitols are used with the pH of 5.4. So here two different enzyme mixtures that will be used, and again these material that will be filtrate out. So the other extra material like debris or other cells, all that will be removed with the help of this enzyme. So these filtrate again they will be centrifuged, so we can got the clear protoplast solution, and this protoplast solution we can store it in the sucrose solution. So these are the two enzymatic methods. First one is the single enzymatic method, and second one is the double enzymatic process. So by using this process, we can easily remove the protoplast from plant cells. Then third one is the applications of protoplast culture. So this protoplast mostly used for the somatic hybridization. That means when we can making the cross in tissue culture. That means when we can use the two protoplasts or two different protoplasts and making the hybrid one. So on that, there is a protoplast technique that will be used for somatic hybridization in laboratory. We can we can make the cross between two different protoplast and got the hybrid protoplast. Then its protoplast culture are also applied in the productions of the gene manipulated plant. So genetically modified जो plant prepare किए जाते हैं. In that technique also the protoplast technique that will be used. Then for getting the disease resistant plants, these protoplast culture are also applied. So these are regarding the methods that have been used for separate out the protoplast. Then the next question is the process of protoplast culture or describe the techniques of plantlet generate from the protoplast. So here हमने देखा पहले क्वेश्चन में कि जो प्रोटोप्लास्ट है प्रोटोप्लास्ट कैसे सेपरेट किया जाता है. So first of all you have to write here about the protoplast. What is protoplast? What is protoplast?
Plant cell without cell wall.
Right. Plant cells without cell wall. So right here about the protoplast. Then the isolation methods of the isolation of protoplast. In that there are two methods: mechanical methods and enzymatic methods. Then enzymatic method. There are two methods: single enzymatic methods and double enzymatic same method. So you can write here same from question 1. Then the process or techniques that have been used in protoplast culture. So first of all, the procedure of the protoplast separation. Protoplast separate कैसे किया जाता है उसके बारे में आप फ्लो चार्ट यहाँ पे है. तो पहले लिप मटेरियल सिलेक्ट किया जाता है, उसे स्टेरिलाइज किया जाता है. स्टेरिलाइज करने के बाद जो भी लीफ है लीफ का पील ऑफ किया जाता है. Then जो mesophyll tissue that will be cut into the small segment. Then they undergoes the plasmolysis. In plasmolysis, there is the cell wall will be or the protoplast of the cell they will be shrink out. So we can easily remove out the cell wall by using the mechanical or enzymatic method. So here using the enzymes or some by using the microscope or scalpels remove the cell wall. Then these mesophyll tissues that will be filtrate out. So due to the filtration, the extra material that will be removed out. Again that filtrate they will be centrifuged, so we can got the pure protoplast. So this is the hub procedure. There is the separation of the protoplast. Then there will be fusions of the gene mutation or addition. That means the in protoplast culture we can directly use this protoplast for the somatic hybridization, or sometimes in these protoplasts some another genes that will be manipulated. So manipulation of protoplasts are also used in tissue culture. And so here these protoplast of the cell organelles, in that we can use directly protoplast, or sometimes the manipulation in the protoplast that will be takes place. So after this protoplast, there will be used in the tissue culture. So these protoplasts that will be kept on the incubation on MS medium. So after the incubation, in controlled condition, these protoplasts form the callus structure. So this callus, again, they will be recultured on new nutrient medium or new slant by using the BAP medium with 1 μm concentration. So there will be shoot initiation will be takes place. Then again the media that have the concentration of cytokinin, then there will be root initiations will be takes place. And after the incubations, the these callus they will be form the plantlets. Again, these plantlets that will be hardening first of all at the laboratory, and then greenhouse, and then in the field. So in this way, these from the protoplast we can got the number of plantlets. So this procedure that will be same like the other method that will be used in tissue culture, but here the first of all the separation of the protoplast that will be very important, and then this separated protoplast that will be directly used for tissue culture, and sometimes make the hybridized or manipulated some another genes on that protoplast, and then they can be grow on the slant. So here we can got the number of plantlets from the protoplast. So in case of this gene manipulation, here some the genes like the NEF genes, some disease resistant genes or high yielding genes that will be we can manipulated in this protoplast, because protoplast membrane that will be permeable. They can absorb these genes. So if we separate out the protoplast, then they can be easily, it is possible to easily inserted these genes like disease resistance, high yielding genes or NEEP genes. So in this somatic hybridization, the gene manipulations of the protoplast are used. Then in case of the protoplast culture, after the isolation of protoplast, it may be used for somatic hybridization or manipulation or induced mutation, cell organelles addition, etc. So this protoplast we can use as a explant, and here this protoplast we can easily treated with the different task. Like in somatic hybridization that means the fusion of the protoplast. So here the protoplast of the same plant or different plants we can select, and they are forced to fuse together. Then the fusion of this protoplast will be carried out by mechanically as well as in chemical methods. मैकेनिकल मेथड बाय यूजिंग द माइक्रो कैपिलरीज। माइक्रो कैपिलरीज का जो एक सेल है, डिफरेंट प्लांट्स के हम प्रोटोप्लास्ट सेपरेट कर सकते हैं, और जो मटेरियल है वो एक ही, जो दोनों जो प्लांट्स है जो डिफरेंट प्रोटोप्लास्ट है उससे हम थ्रू माइक्रो कैपिलरीज एक जगह पे कर सकते हैं। देन जो केमिकल मेथड है, केमिकल मेथड में बाय यूजिंग द हम केमिकल लाइक द पॉलीइथिलीन ग्लाइकॉल और यूज कर सकते हैं। दिस पॉलीइथिलीन ग्लाइकॉल इट इज आल्सो कॉल्ड एज फ्यूजोजेन। फ्यूजोजेन that means they can fuse the two different protoplasts. So by using these methods, mechanical as well as chemical method, we can fuse the protoplast, and due to this fused protoplast we can got the somatic hybrid plants. देन जीन मैनिपुलेशन का मैंने बताया, जीन मैनिपुलेशन में जो प्रोटोप्लास्ट मेम्ब्रेन है, प्रोटोप्लास्ट मेम्ब्रेन परमिएबल रहते हैं, they can easily absorb these genes. So here we can use the different genes like disease resistant genes. ये disease resistant genes हम वहाँ पे रखेंगे तो जो प्रोटोप्लास्ट है इजीली वो जीन्स एब्सॉर्ब कर सकता है। सो जो न्यू वैरायटीज, जो न्यू प्लांटलेट्स होंगे वो कैसे होंगे, डिसीज रेजिस्टेंस वहाँ पे मिलेंगे। तो इन दिस वे वी कैन इजीली मैनिपुलेटेड द कमर्शियली इम्पोर्टेंट, जो भी कमर्शियली इम्पोर्टेंट जीन्स है वो हम इजीली प्रोटोप्लास्ट में इंसर्ट कर सकते हैं। Then induction of the mutation in protophase, that will be also carried out by using the different mutagens. Here we can directly use the mutagens, so we can manipulate or induct this protoplast. Then there is also possibility of the addition of the cell organelles in protoplast. In cell organelles like mitochondria or chloroplast carry the additional gene. So for antibiotic resistance, then fermentations, or uh, we can easily uh, added out some cell organelles in protoplast. Then to create the variations or the extra chromosomal characters, we can use this protoplast. So with the help of this protoplast, we can also added out some genes that will be beneficial at the commercial level. So here we can add these genes or different manipulations or mutation that will be carried out on this protoplast. So commercially it will be very helpful. Then after this there will be प्रोटोप्लास्ट कल्टीवेशन। सो इन प्रोटोप्लास्ट कल्टीवेशन जैसे नॉर्मल हम टिश्यू कल्चर प्रोसीजर यूज करते हैं, सेम लाइक हियर वी कैन यूज द or we can go through the somatic hybridization or gene manipulation or induced mutation or adding some cell organelles into the protoplast. Then this protoplast that will be inoculated on the MS medium. Then this slant that will be kept in incubation. So in incubation, after the formation of the callus, we can remove this callus again recultured onto the new slant and kept for in control condition. Then there will be formation of the root and shoot that will be takes place, and after the formation of the plantlet they will be undergoes for the hardening. So in hardening, first of all at the laboratory and then on the field, hardening will be goes on. So in this way, the different hybridized as well as some manipulated or uh, resistant variety we can got by using this protoplast culture. The applications of this same like the last one question. So here the process of the protoplast culture will be takes place. So here it will be long question, but first of all you have to write about the protoplast, then methods that will be used for protoplast separation, then the protoplast incubation or protoplast method that will be used in tissue culture. You have to write in details. And last one is about the applications of protoplast culture. So here the questions will be over about the protoplast culture.